Hello, student. A very good afternoon to all. Okay. So my audio is clear and my screen is visible. Good afternoon, Nisha. So today we are going to discuss body fluids and circulation. So as you all know that the body fluid, they are help in transporting certain substances all over the body. And these certain substances, they could be nutrients, they could be solutes, it could be hormone, and it could be an excretory product as well. So there are two types of fluid which are mainly important. One is blood and the second is lymph. Blood is a special connective tissue and it is mesodermal in origin. Please remember the origin. Blood is mesodermal in origin. It consists of fluid matrix, plasma, and formed elements. Formed elements consist of RBC, WBC, and platelets. And plasma, it consists of mainly water, some solutes, and proteins. Major composition is of water in case of plasma. Now, the blood vessels, they are composed of arteries, veins, and capillaries. Arteries, they usually carry the oxygenated blood, except the pulmonary artery, which carry the deoxygenated blood. Veins, they usually carry deoxygenated blood, except the pulmonary vein, which carry the oxygenated blood. And capillaries, they are thin and they have a single layer of endothelium. These capillaries, they help in forming a close network between the arteries and the veins. Then they are synthesized in the red bone marrow. This is the bone marrow. This portion is the red bone marrow. This is also red bone marrow, and the middle portion is yellow bone marrow. So the red bone marrow, they give rise to, they consist of stem cell, and these stem cells, they form RBC, WBC, and platelets. One more important point, beta, these platelets, they are formed by the cellular fragmentation of megakaryocyte. So first stem cell will produce megakaryocytes. These megakaryocytes, they undergo cellular fragmentation, and because of that cellular fragmentation, small platelets are formed. And these platelets, they play a very important role in blood clotting because they contain thromboplastin. Next is the lymph. This is blood. If I remove RBC plus large proteins, and blood platelets, if I remove RBC, large proteins, and blood platelets from the blood, the rest part is known as lymph. Okay? I have a less space. Let me draw it here. This is the blood vessels. Okay? These are the capillaries. Again, a blood vessel. When the blood flows through the blood vessels, when it reaches the capillaries, some of the part from the blood, they diffuse out of the capillaries. These are water plus water-soluble substances. These are diffuse out. And these substances, component, the water and the water-soluble substances, they get accumulated in the space between the cells of the tissues. And this is what we call a tissue fluid. These get diffused out, and these are the cells, space between the cells, they get accumulated here, like this. This is what we call a tissue fluid. And with this tissue fluid start moving into the lymphatic vessels, we call it as lymph. Okay? And it helps in the transport of nutrients and hormone. The fat are absorbed. The fat, these are absorbed like a chylomicron. Okay, chylomicron, they are the fat globules surrounded by the protein, and they move into the lymphatic vessels, which is known as a lacteal in case of villi. Moving next is the component of blood. One is plasma, which constitute 55%, and formed elements, which constitute 45%. So plasma constitute the major portion of the blood, that is 55%. And the components of plasma is water, which is 90 to 92%, minerals like sodium, calcium, chlorine, potassium, etc., nutrients like glucose, amino acid, lipids, and protein. This is important beta. Remember, proteins are albumin. Albumin is large in number. That means they are abundant in plasma. Second, they maintain osmotic balance of the blood. Remember this point. Second is fibrinogen. Fibrinogen is a clotting protein. It plays an important role in clotting. The blood clot which is formed during the injury. Globulin is alpha globulin, beta globulin, and gamma. These alpha and the beta, they are produced in liver, but these gamma, they are produced by plasma B cells. And these plasma cells, and these gamma, they are immunoglobulin like IgA, IgM. They have a mechanism in defense. They play a very important role in defense. Formed elements, it consists of, I already told you, they consist of RBC, WBC, and platelets. Like RBC, WBC, and platelets, RBC constitute the major portion, that is 5.5 to 5.5 million mm³. They does not have nucleus, they are enucleated. That is why there's a maximum space for hemoglobin. And they have a very short lifespan, which is 120 days. WBC, they are known as leukocytes. They are 6,000 to 8,000 mm³. They are colorless because it does not have hemoglobin. Nucleus is present, important. In RBC, mature RBC does not have nucleus, but WBC have nucleus. The nucleus is present. And on the basis of that, they can be neutrophil, which are multi-lobed. Okay, they can be eosinophil, which have headphone-like nucleus, basophils, which having a S-shaped nucleus. And they are lymphocyte as well and monocyte, which we will discuss in the next slide. Now they have a lifespan is 1 to 15 days. They also have a very short lifespan. Platelets, they are 1.5 to 3.5 lakh mm³ in number. They are colorless. They have a nucleus absent. So very important point, beta. The nucleus is also absent in platelets, and it is also absent in RBC. Their lifespan is 1 week. And these platelets, they are also known as thrombocytes, and they contain thromboplastin, which play a very important role in blood clotting. Moving next is the type of white blood cells. White blood cells are of two types: granulocytes, which contains granules, or where there's a presence of granules. Granule present. Here the granules are absent. That is why they are agranulocytes. Granulocytes are of three types: neutrophil, eosinophil, and basophil. Neutrophil, they are constitute 60 to 65% of the leukocytes, and they have multi-lobed nucleus. You can see that there's one lobed here, one lobed here, one here. They're having a 3 lobes. There is a multi-lobed nucleus. And these short, these are granules. Okay, here also granules are present. In basophils also granules are present. That is why they are granulocytes. And they help in phagocytosis, or they are phagocytic in nature. That means they have the ability to engulf the pathogen or the foreign particles which has entered the body. Eosinophils, They have headphone-shaped nucleus like this. They have headphone-shaped nucleus. Okay, remember, they release antihistamine or histamidase. Basophil, they are largest in number is neutrophil. Basophil is 0.5 to 1%. This is less in number. Okay, and they have S-shaped nucleus. And the basophil, they release histamine and heparin. Okay, next come is the lymphocyte. Lymphocyte, they generally have a very round nucleus. And the agranulocyte, that is why you can't see any granules, where the granules are present in these neutrophil, eosinophil, and basophil. But in lymphocyte, there's no granules. So they have a round nucleus. That is why they agranulocytes. They are of three types. They can be a B lymphocyte, T lymphocyte, and natural killer cell. B lymphocytes, they produce plasma B cells and the memory cells. Similarly, T lymphocytes produces T helper cell, T cytotoxic cell, there's T suppressor cell, and memory T cell. Okay. Next is monocyte. Monocyte, they are large in number. They are large in size. Sorry. They have a kidney shape, bean shape, and horseshoe shape nucleus. And they are phagocytotic in nature. Okay. Moving next is the coagulation in blood. That is, now we are going to discuss the role of platelets. When the tissue get injured, okay, injury has taken place on tissue. Tissue will produce thromboplastin. Now this thromboplastin, okay, now this thromboplastin will attract more platelets. Now these platelets, they are also thrombocytes, right? They're also known as thrombocyte. They also have thromboplastin. They will secrete thromboplastin. Now this thromboplastin will activate thrombokinase enzyme. Now this thrombokinase will convert prothrombin to thrombin. And calcium play a very important role here. Please remember calcium. Now this will convert to thrombin. Thrombin will activate fibrinogen, will act on fibrinogen, and the fibrinogen will convert it into fibrin. This is the protein or the fibrin which play a very important role in clotting. It will lead to clot. You can see the fibrinogen converted to fibrin, and therefore fibrin, they form fibers long. These fibrin fibers, they trap the RBC and result in the blood clot. I hope it is clear. Moving next is the ABO blood grouping and RH blood grouping. So RBCs, they have self-antigen. Self-antigen are A and B. These are the self-antigen. Okay. If the person is having a blood group A, this person will have A antigen on the surface. And this person will have antibody B. Because for this person, the antigen B will act as a foreign particle. So this person will already have an antibody B against that antigen. But the self-antigen is antigen A in case of person which is having a blood group A. Okay. And if this person has Rh factor on the surface of RBC, if this person have Rh factor, then we call this person having a blood group A positive. And those persons which are A negative, they don't have this Rh factor on the surface of RBC. They will have antigen A, but they won't have Rh factor. That is why they are A negative in blood group. Okay. And remember one important point. If this person who is having a blood group A will receive a blood, can only receive a blood from A or O. It cannot receive the blood from B or AB because B will have antigen B and this person will have antigen B already. Sorry, this is a blood person having a blood group A. So this person will have antigen A on their surface, right? And if it receives the blood from the person B, that person B will have self-antigen is antigen B, but antibody is A. So this antigen A and antibody will combine and this leads to a clumping of blood. So that is why the person having a blood group A will only take the blood from A and O, cannot take from B or AB. So on the basis of these, we can say that AB is a universal recipient because it has antigen A and antigen B. It does not have antibody. It does not have any antibody. Okay. So it can receive the blood from B, A, and O because A does not have antigen. And A is a universal donor because it does not have antigen. Remember this. Moving next is the Rh incompatibility. So what happens if the mother is Rh negative and the fetus is positive? Okay, during the first pregnancy, is a mother fetus. This is a barrier. This is a barrier between the mother and the fetus, and the barrier is the placenta. Okay. So during the first pregnancy, the mother, the Rh negative mother, and this is fetus. So there's no mixing of blood takes place between the mother and the fetus because there's a barrier, which is the placenta. But during the first delivery, this placenta breaks and the maternal blood get exposed to the fetus blood, because of which the mother develops the Rh antibodies. These are the antibodies, okay? So now the mother has developed Rh antibody. So during the second pregnancy, if the fetus is again Rh positive, then the mother already has the Rh antibodies, and these Rh antibodies will enter into the fetus and they will destroy the RBC of the fetus and leads to hemolytic disease. Hemo means RBC and lytic is lysis. So they will break down the RBC of the fetus, and this leads to erythroblastosis fetalis. So generally after the first pregnancy, the mother, they are given anti-Rh antibodies. What happened that these anti-Rh antibodies will destroy these antigen, Rh positive antigen, and does not allow the mother to form the memory bodies. It does not allow the mother to form the memory antibodies. Okay. So this is the role of anti-Rh antibody. Moving next is a type of circulatory system. There are two types of circulatory system: open circulatory system and closed circulatory system. In open circulatory system, the heart pumps the blood passes through the large vessels and it reaches to the open spaces or sinuses. You can see that there's this open space. So this type of open circulatory system is found in arthropods, in non-cephalopod mollusk, in tunicata, which is Urochordata, Hemichordata, and Echinodermata. Closed circulatory system when the blood pumped by the heart is always circulated through a closed network of blood vessels. Okay, there's no open space. There's a closed network of blood vessels. Okay. And one more important thing, that in open circulatory system, the capillaries are absent. In closed circulatory system, the capillaries are present, because of which they're forming a closed network of blood vessels. Moving next is the type of blood vessels. There are three types. There are arteries, there are vein, and there are capillaries. Capillaries are very thin and they have a single endothelium layer. Okay, they have a single endothelium layer. Arteries, you can see they have a very narrow lumen. Or the lumen is, diameter of lumen is small. They have three layers. Outer is tunica externa, middle is tunica media, and inner is tunica intima. Okay, this is the inner layer. In case of arteries, the tunica media is thick. You can see this, it is thick. Because in arteries, the pressure is high. Why the pressure is high? Because the blood from the left ventricle is pumping to the aorta, which carry the oxygenated blood to the rest of the body. So arteries, they usually carry the oxygenated blood and they have a high pressure. Because of high pressure, they does not have a wall. Okay, and the arteries, they carry the blood away from the heart and they always carry the oxygenated blood. Except the pulmonary artery. Remember this. Moving next is the vein. Vein, you can see it have a very large lumen. It also have three layers like tunica externa, tunica media, and tunica intima. They have valve. The pressure in vein is low. Because the pressure is low, so they have a presence of valve to prevent the backflow of the blood. Vein, they carry the blood towards the heart, and all the veins, they carry deoxygenated blood except the pulmonary vein, which carry the oxygenated blood. Remember this. Moving next is the heart, which is very important, okay? Heart is mesodermal in origin. Remember this. It is located between the lungs and tilted towards the left. There is superior vena cava. Superior vena cava, it received the deoxygenated blood from the upper part of the body and transferred it to the right atrium. Okay? So it will collect the blood from the upper part of the body and transfer all the deoxygenated blood into the right atrium. All the blood will collect in the right atrium. Next is, there's also one inferior vena cava, remember. There's superior vena cava and there's inferior vena cava. Inferior vena cava will receive the blood from the lower part of the body. It will receive blood from the, it will collect the blood from the lower part of the body and transfer it into the right atrium. So right atrium will receive the deoxygenated blood from the superior vena cava and from the inferior vena cava. And inferior vena cava, it also have a wall to prevent the backflow of the deoxygenated blood. And that wall is known as a Thebetian wall. Sorry, that wall is known as a Eustachian wall. Thebetian wall is present in a coronary sinus, which is a cavity present in the right atrium. And remember one point, beta, there's a septums are also present. And these septums, they are like with the septum between the atrium are known as interatrium septum, and the septum present between the ventricles are known as the interventricle septum, and the septum is also present between the atrium and the ventricle. They are known as the atrioventricle septum. Okay, so there are 3 septums are already present. Remember this. So the valve which is present in the inferior vena cava is the eustachian valve. And I've already told you about the septum. Now this deoxygenated blood from the right atrium will enter into the right ventricle. Now the deoxygenated blood has reached the right ventricle. Okay, and there's a valve is present. This is a valve. The wall is known as, in case of right atrium, the wall present between the right atrium and the right ventricle is known as a tricuspid valve. It's also known as a AV valve, which is avioventricular valve. And the function of this valve is to prevent the backflow of the deoxygenated blood. Okay. Now this deoxygenated blood will move into the pulmonary artery, and pulmonary artery will carry this deoxygenated blood to the lungs. Okay, and there's also a valve present in a pulmonary artery which is known as a semilunar valve. Okay, remember the name of the valve and their location. They are very important. Now the deoxygenated blood has reached the lungs. Now the lung, where the exchange of gases will take place, and now this oxygenated blood from the lungs will be carried by this pulmonary vein, which carry the oxygenated blood and will reach to the left atrium. Now the oxygenated blood has reached the left atrium. Okay. And there's also a valve is present between the left atrium and the left ventricle, which is known as a mitral valve, or it is also known as a bicuspid valve. Okay. Also known as a bicuspid valve. Now this oxygenated blood from the right atrium will enter into the, from the left atrium, now this oxygenated blood from the left atrium will move into the left ventricle. Now all the oxygenated blood has reached the left ventricle. Now this oxygenated blood will enter into the aorta, and aorta will carry the oxygenated blood to the rest of the body tissue. So here also the valve is present in the aorta, which is known as a aortic valve, or is also known as a semilunar valve. Okay, and please remember the walls of the left, you can see this, the walls of the left ventricle, they are thick as compared to the right ventricle. Why? Because the left ventricle, it has a lot of pressure because it has to pump the oxygenated blood to this aorta, and aorta has to carry the oxygenated blood to the rest of the body. So left ventricle has thick wall as compared to the right ventricle. I hope it is clear. Now the conduction of heart. As you know that heart is myogenic in nature. Myogenic means that the heart can initiate its own impulse. It does not require the information from the brain to generate the impulse. And because why it can generate its own impulse? Because it has the presence of nodal tissues. These nodal tissues, they are auto-excitable. Okay, and these are SA node, they are AV node, they are AV bundle, which are also known as a bundle of His, and there's a Purkinje fibers. These are the 4 nodal tissue, okay? First, we are going to discuss the SA node. Which is also known as a sinoatrial node, or the pacemaker, because it can initiate the heartbeat. So it is a pacemaker. It generates an impulse of 70 to 80 impulse per minute. Okay, so position of SA node is present at the top of the right portion of the right atrium. Okay, and this SA node, the impulse generated by the SA node will move to the left atrium and also received by the right atrium. So when this impulse is received by the left atrium and the right atrium, what happens? The atrium depolarization occurs. Depolarization because the membrane potential has changed. So depolarization has occurred, because of which the atrium contract, which lead to contraction of the atrium, which we call it as atrium systole or atria systole. Okay. SA node also passes the impulse to AV node. Okay, it also passes the impulse to AV node, and when the AV node receives the impulse, it will transfer to the AV bundles. We also call it as bundle of His, which are present in the intraventricular septum. Remember this AV bundles, they are present in the, this is the septum present between the two ventricle. It is known as the interventricular septum. So these AV bundle, they are present in the interventricular septum, and they divide and they form a right bundle and the left bundle. Okay, and these AV bundle, they give rise to Purkinje fibers, and these when the signal from the the impulse from the AV node reaches to the AV bundle, from the AV bundle the impulse reaches to the Purkinje fibers, and Purkinje fiber leads to the ventricle depolarization, the ventricle wall depolarized. When the ventricle wall depolarized, the ventricle, it leads to the contraction of ventricle, and when the ventricle contract, it leads to the ventricle systole. Okay. Moving next is the double circulation. Double circulation, it consists of pulmonary and systemic. Pulmonary means right side of heart. This is taking place right side of the heart. Systemic is left side. So in pulmonary, what happened? The deoxygenated blood from the right ventricle reached to pulmonary artery, then lungs, and now the oxygenated blood will be carried by the pulmonary veins and reaches to the left atrium. Remember this, it started from the ventricle and reaches to the atrium. Similar in systemic also, starting from the ventricle and reaches to the atrium. Okay, in systemic, what happened? The oxygenated blood from the left ventricle carried by aorta, it reaches to arteries, then arteriole, then body tissues, then deoxygenated blood from the tissue with venules, then veins, vena cava, and finally to right atrium. Okay, so this is a complete double circulation. Hepatic portal system, hepatic means liver beta. Remember, hepatic always means liver. So hepatic portal system is a type of a special, let me draw it here, is a type of a special capillary network between the two organs, like small intestine. Small intestine, there's a vein, then this is the liver and its heart. This is a hepatic portal vein, which carry the blood from the small intestine to liver. Okay, and this vein carry the ammonia-rich blood. When this ammonia-rich blood reaches to liver, where the Krebs-Hensley cycle occur, and this ammonia is converted to urea. And the vein which connects the liver and the heart is hepatic vein. Okay, so this is a hepatic portal system which is connecting the two organs. Next comes the coronary circulation, is the circulation which is taking place in the heart. Okay, just give me a second, let me just draw it. So in coronary circulation, what happens? Aorta will carry the oxygenated blood. Okay, oxygenated blood will be carried by the coronary artery. This coronary artery will transfer the oxygenated blood to heart muscles. Heart muscles receive the oxygenated blood. Now this coronary vein will carry the deoxygenated blood from the heart muscles. Coronary veins will give the deoxygenated to coronary sinus, which is, I told you, it is a cavity or the space in the right atrium, and coronary sinus will give the deoxygenated blood to right atrium. So this is a circulation in the heart. So it is a coronary circulation. Okay, that circulate the blood to and from the cardiac muscles. Moving next, this picture shows you a double circulation, the pulmonary circulation and the systemic circulation, which I've already told you. Hepatic portal system, can see these hepatic portal vein, which are connecting the liver and the small intestine, and this is the coronary circulation. This is a type of circulation. This portion shows you the joint diastole. At this point, the 70% blood fill in the ventricle. So ventricle in this stage is the diastole stage. Diastole means relaxed. Okay, so at this point, when the ventricle is diastole or when the ventricle is relaxed, 70% of the blood is filled. Okay, now atrial contraction takes place. Remember, the SA node will give the impulse to the right atrium and the left atrium, which lead to the atrium depolarization. And polarization, because of the depolarization, where atrium contraction occurs, which we call as atrium systole. So atrium systole begins and it lasts for only 0.1 second. This is for 0.4 second, the joint diastole. Okay. When the atrium contract, that means they contract, they squeeze out of the atrium. So that rest of that means all the blood which in the atrium will move to the ventricle. Now, remaining 30% of the blood fill in the ventricle. So it leads to 100% filling of the ventricle. Now the ventricle is filled, 100% filling, okay? At this point, the portion between the atrial contraction and ventricle contraction, at this point, the sound lub is produced. The first sound which comes out is lub. Why? When this atrium squeeze and contract, all the blood from the atrium will reach to the ventricles. Okay, it will reach to the ventricles, then the walls, the AV walls, the dicuspid wall and the tricuspid wall, which are present between the atrium and the ventricle, they get closed. And when they close, they produce a sound which is lub. At this point, these walls, the AV wall, they get closed, but the semilunar wall of the pulmonary artery and the aorta opens. And when the signal, when the SA node transfer the impulse to AV node to AV bundle, from the AV bundle to Purkinje fibers, which leads to the ventricle depolarization and the ventricle contraction or the ventricle systole. Now the ventricle will squeeze or contract. When the ventricle contract, all the blood from the ventricle, they will move to the pulmonary artery or the aorta. The oxygenated blood will move to the aorta and deoxygenated blood will move to the pulmonary artery. Okay, at this point, sound dub is produced. Why? Because now when this pulmonary artery and the aorta receive the blood, it filled with the blood. Now to prevent the backflow of the blood, the semilunar wall, the valve of the pulmonary artery and the aorta, they get closed. At this point, the sound produces dub. Okay? And this ventricle systole require a time of 0.3 seconds. So the cardiac cycle, entire cardiac cycle requires 0.8 second time. This is the phases of cardiac cycle which I have already discussed, that when the atrial systole begins, the atrial contraction forces the blood into the ventricle. You can see the blood from the ventricles, from the atrium, the blood are moving into the ventricles. This happens when the atria depolarize or the atrium contraction or the atrium systole takes place. Now the ventricular contraction pushes the AV wall closed. Now when the ventricles are filled, they put a pressure, and these AV walls, they get closed. Here these are open, but now at this point they are closed. And this happens because of the high pressure in the ventricles. And this wave, this QRS wave, is known as a ventricle depolarization. At this point, the ventricle depolarization occurs and the ventricle contract, and which put a pressure on the wall, and the wall get closed. Now the semilunar walls opens and the blood will eject out. Now the blood will move to the pulmonary artery and the aorta. Now the semilunar wall closed and the blood flows into the arteria. Now see, these walls are closed. These semilunar walls are closed, and here the sound produces duh. Okay? Ventricular diastole, which is the late phase. Here what happens, the chamber relaxes and the blood fills ventricle passively. Now moving next. Now we have very less time. So cardiac cycle is completed in 0.8 seconds. One heartbeat equals to one cardiac cycle, and normal heartbeat is 70 to 75 times per minute. Stroke volume. Stroke volume is the amount of blood which is ejected from the ventricle, okay, per beat. For example, like bottle. Okay, if a bottle is filled with water, if I squeeze that bottle, some water will come out and some will remain in the bottle. Okay, some water will remain in the bottle, but some water will come out because I have squeezed that bottle in the middle. Similarly, the amount of blood which is ejected by each ventricles per beat is known as stroke volume, and it is about 70 ml. Cardiac output is the volume of the blood which is pumped out of each ventricle per minute. Remember, each ventricle per minute is cardiac output, per cycle is stroke volume. And cardiac output is calculated as stroke volume into heart rate, which is 5,000 ml of 5 liter. So there's a machine called electrocardiograph. This machine measures the electrical activity of the heart, and the graph which is produced is known as electrocardiogram. Okay, so this shows you a wave. This is a P wave. P wave represents the atrial depolarization. Remember, QRS wave represents ventricular depolarization, where ventricle systole happens. T wave represents the ventricular repolarization. Better remember these waves and the event which is taking place at each wave. They are very important. The first sound which is produced is lup, and the second sound which is produced is dove, which I have already told you. Lup produced when AV wall get closed and semilunar wall open. Dove produced when semilunar wall get closed. Okay. Moving next is the regulation of heart. Generally, the heart is regulate. Okay, sorry. Generally, the heart is myogenic in nature. That means it can regulate its cycle on its own. But sometimes brain also play a important role. Brain can also regulate the cardiac cycle. For example, there's medulla oblongata, which is a part of CNS. There's NAS, which is autonomic nervous system. And this ANS has two parts, the sympathetic and parasympathetic. Sympathetic, when act on SA node, it releases epinephrine and norepinephrine. And these two will act as a neurotransmitter, and they will increase the heart rate as well as the cardiac output. Parasympathetic is this system is active when we are in relaxed state. Okay. So when parasympathetic is active, it acts on SA node. Then parasympathetic will release acetylcholine. This will decrease the heart rate as well as the cardiac output. Remember one point, that heart is generating its impulse on its own, but these cardiac and heart rate, cardiac output and the heart rate can also be regulated by the brain. Moving next is the disorder, cardiac arrest, complete stoppage of heartbeat, heart failure, when the heart is not pumping blood effectively to meet the need of the body. Atherosclerosis or the coronary heart disease is occurred due to the deposition of the calcium. There's cholesterol, fat, or fibrous protein into the arteries, because of which the lumen of the artery get narrow and the enough blood cannot reach the heart. High blood pressure. Beta, blood pressure is the pressure which is exerted by the blood on the walls of the arteries. Remember, this is what we call pressure. Okay, and if the person is having a diastolic and systolic pressure of 140, systolic and diastolic pressure is 140 and 90, that person is having a high blood pressure, hypertension. And high blood pressure leads to affect the vital organs like brain and kidney. Then the angina pectoris, which is acute chest pain. It happens when not enough oxygen is reaching to the cardiac muscles. Heart attack, when the heart muscles is suddenly damaged by the inadequate supply of blood. Remember these are very important, okay? All of these. Sometimes the direct question came from the disorder of circulatory system in NEET, okay? Moving next is the question. Question number 1 says inadequate supply of oxygen to heart muscles leads to symptoms of acute chest pain. This I already just now told you. This order of the circulatory system is identified as cardiac arrest, heart failure, coronary heart disease, or angina pectoris. What do you think? What will be the correct option for this question? Quickly, everybody. It's already 12:50. Remember, acute chest pain. Okay. Very good, MS Gauri. Very good, Dhruvi. Yes, so the correct option is angina pectoris. Cardiac arrest. Cardiac arrest is when the heartbeat stop, like complete stoppage of the heart. Coronary heart disease, when there's a deposition of calcium, cholesterol, fat, fibrous proteins in the blood vessels, because of which the atrium lumens narrow. Angina pectoris is the acute chest pain when not enough oxygen is reaching to the heart. Heart attack, when the heart muscles is suddenly damaged by the inadequate supply of blood. Remember all these points. So the correct option for this is angina pectoris, acute chest pain when enough oxygen is not reaching to the heart muscles. Next, question number 2 says a specialized nodal tissue embedded in the lower corner of the right atrium close to the atrioventricular septum delays the spreading of impulse to heart apex for about 0.1 second. The delays allow. Quickly answer. What do you think? What will be the correct option for this question? Yeah, this is the right atrium. Okay. This is SA node, which is present top right portion of the right atrium. And here the AV node are present, which is present lower portion of the lower corner of the right atrium and close to the atrioventricular septum. That is the septum between the atrium and the ventricle is known as the atrioventricular septum. So they are talking about this AV node. Okay. So a specialized nodal tissue which is embedded in the lower corner of the right atrium, that is lower corner of the right atrium is AV node, close to the atrioventricular septum, delay the spreading of impulse to the heart apex. This is the apex of the heart and this is the base. Remember this. For about spreading of impulse to heart apex for about 0.1 second. The delays allow the atrium to empty completely. Okay, यहाँ पे ये process delay किया है, the spreading of impulse to the heart apex, that because the AV node transfer the impulse to the AV bundle which are present in the interventricular septa, and the interventricular septum will give the impulse to the Purkinje fiber. Here these Purkinje fibers are present. So they are delaying these signals to AV bundle, because if the signals are transferred to, or the impulse are transferred to AV bundle will transfer the impulse to Purkinje fiber and it will lead to the contraction of ventricles. So this is delaying the contraction of ventricles and allow the atrium to completely empty, so that when atrium systole takes place, all the blood from the atrium will enter into the ventricle and the ventricle are completely filled. Okay, this blood, option 1 says blood to enter aorta, ventricles to empty completely, blood to enter pulmonary arteries. These all are related to ventricle contraction. When ventricle contraction occurs, then the ventricle contract or squeeze the blood from the ventricle, they will move to the deoxygenated blood will move to the pulmonary artery, and the oxygenated blood will move from the left ventricle will move to the aorta. Okay, so the correct option for this is atrium to empty completely. I hope it is clear to everyone. Moving next, what would be the heart rate of a person if the cardiac output is 5 liter? Blood volume in the ventricle at the end of this diastole is 100 ml and at the end of ventricular systole is 50 ml. Okay. First is end of diastole. End of diastole is 100 ml. End of diastole means the amount of blood which is present in the ventricle at the end of the ventricular diastole. So here it is 100 ml. Now end of systole, it is given 50 ml. End of systole means the amount of blood which is present in the ventricle at the end of the ventricular systole. For example, like bottle. If I squeeze that bottle, some water will come out and some will remain in the, some water will come out and some water will remain in the bottle. Okay. So now the stroke volume will be 100 minus 50. It will be 50 ml. Okay, stroke volume will be 100 minus 50 equals to 50 ml. Here the cardiac output is given, which is 5 liter. And we know that cardiac output equals to stroke volume into heart rate. Okay, they're asking about the heart rate of a person. Cardiac output is 5 liter. I convert it into ml, which is multiplied by 1000. Now it is 5000 ml. Okay. Stroke volume is 50 ml. Heart rate will be 100 beats per minute. Correct option would be 3. Very good, Chechuni. It is 3. Very good, beta. I hope it is clear to everyone. Now it's already 1. We are winding up the class. Thank you all for your participation. I hope this lecture was informative for you. Please try to do more and more previous year questions. They are very important. Thank you all and see you in the next class. Have a nice day.